Hi, so really quickly in this video, I want to talk a little bit about nuclear pore structure. And so we're going to specifically talk about sort of this complex right here. So in the nucleus of a cell, what we'll have, again, this will be in eukaryotic organisms because they'll have nucleuses. We'll have a membrane right here that goes around the nucleus. And so in here will be inside the nucleus and out here will be outside of the nucleus. And this membrane right here is actually continuous with the endoplasmic reticulum. So these large pores will be used for getting proteins in and out of the nucleus. And really what the effective range of size of proteins that can go in and out of the nucleus are above 25 kilodaltons. It actually becomes pretty difficult for them to freely diffuse in and out of these pores. And above 40, it's pretty much impossible, so they would need to be actively transported in that case. But this membrane right here blocks pretty much everything, all proteins, and then this pore is where all of those proteins that can diffuse through are going to diffuse through. So in this complex, we have these nucleoporin proteins, and so these proteins are what are going to sort of check what should or should not go in here and facilitate the right sort of diffusion. We've got a nuclear basket and cytosolic fibrils, and what these will do is cytosolic fibrils will actually sort of latch onto proteins and increase the in this area over here in the cytosol. It's going to increase the concentration of proteins, which is going to facilitate the diffusion through the pore. We've got our membrane ring proteins, which go through the membrane and sort of hold everything on here. And the nuclear basket is actually going to have a similar function to the cytosolic fibrils, but going the other way. And you're going to have in here binding sites for certain proteins, so we can facilitate their import and export with special proteins called importins and exportins.